Okay, the next podcast goes into looking at the history of life on Earth. And so we're looking at the past, we're looking at the theory of macroevolution. Mainly what we've been talking about up to this point has been microevolution. Excuse me. And we've made some references to macroevolution. But macroevolution is the larger picture about what has happened in the past, and it is a theory that organisms change drastically from one type of organism into another. And it is one of the predominant themes about the interpretation of the evidence, but there is different views and theories about that, and we did allude to that in some of the earlier lectures. Um, But in this, one of the first things we look at is the question of where life came from to begin with. So biology is the study of life. That's certainly a question biology addresses. Of course, no one was there at the time when life originated. We don't have any direct observational evidence of this, so we develop theories about this. And there's some different theories. We talk about them in the outline there. And one of the theories is that life arose in the early Earth atmosphere in what's sometimes called primordial soup or primordial mix in the oceans, where chemicals and compounds came together that were floating in this mix to form the first living cell. Now that kind of goes against what we know about the biogenetic law that life comes only from pre-existing life, but that's one theory about how life first originated on Earth. Okay, so that is one theory. Of course, other theories include different types of creation views or intelligent design views, that life is too complex to have happened by pure chance, that it was designed or created by a designer. So those are two perspectives about where life came from. A third one that we mention here, your book mentions, is called panspermia. Panspermia and it was promoted by a very famous scientist, Francis Crick, probably recognize his name, one of the co-discoverers of the structure of DNA, helped to develop, and he promotes this theory. And this theory is that life originated here on Earth from another planet. Um, and his reasoning is that it is just too statistically impossible that life could just happen by pure chance processes and just originate out of chemicals that are floating in the ocean. Just the odds of that happening are astronomically low, and so Crick says that could happen, and he does not like to invoke a designer or creator into an explanation about where life came from. So this is a third um, option that he developed, that life originated somewhere else, and maybe that somewhere else life could come about easier than we know here on Earth. It's not happening, life coming from non-life. And then maybe asteroids or something brought it here to Earth, kind of science fictioning. But those are kind of the only options there for where life came from that got here on Earth, of those three options. Now, this idea that um, life arose in this primordial mix in the oceans on the early Earth was tested. It was tested by um, researcher Stanley Miller. Him and his some of his fellow scientists, Robert Urey, were going to test this idea. Oh, before we get to that, this idea first became developed by Oparin. So Oparin is one that really kind of published this theory that life could happen from non-living stuff in this early Earth. So O'Perrin published that early in the 1920s, I think. O'Perrin published that. But Stanley Miller came along and said, well, I'm going to test this a little bit in the lab. So he built an apparatus. You see this in all biology textbooks. Here's the apparatus. So his apparatus included what Urey said would be in that early Earth atmosphere. He included some gases, some hydrogen, some carbon compounds, water vapor, put it into this atmosphere, put it into this apparatus that was supposed to mimic this early atmosphere O'Perrin wrote about. All right, so what he did is he put those compounds in here, and he put some water vapor in here, heated it up. This is supposed to be mimicking the oceans, the early aquatic environments of the Earth. And then these would condense, these would develop into water vapor, would be carried into this part of the apparatus where the compounds are, and then a spark would be a voltage, a 50,000 volt spark would be ignited inside the apparatus. And so what would happen, the water vapor would collect these parts of these compounds that would be broken by the voltage. Voltage would also bond molecules, help to bond molecules together. So we'd have some mixing of compounds in there. And this is supposed to be mimicking lightning strikes that would hit this early Earth aquatic atmosphere. And then that water would condense and collect down here into a sample for collection. Now Stanley Miller would collect these samples, go look at it under the microscope, and what he found was he found a living cell. No, I'm just kidding. He did not find a living cell. We've never been able to create life in a lab from non-living stuff. So he didn't find that. He didn't prove O'Perrin's theory. But what he did find in this sample was mainly parts of these compounds that were broken that were meaningless. But he did find a few things in here like amino acids. And amino acids are, they're a long way from being a living cell, but they are building blocks for proteins. If you remember back from Bio 1, they're building blocks of proteins, and proteins are certainly essential compounds for a cell, living cell. So could say, and many do, that this gives some support to O'Perrin's theory of life forming from non-living things in this early Earth atmosphere. Now, there's some other things that have to occur for a cell to come into existence. We have our amino acids, they have to accumulate together to form a long chain to form a protein, and it has to be in just the right sequence. Also, we have to have genetic material to get in there. And then all of these things have to come together to form into a cell. So there's some scenarios, and your book uses that term scenario quite a bit with this, because theory implies that we have some direct testing we can do, and a lot of this is, like I say, scenarios about what could have happened. No one was there, and we can't really carry out tests that prove these things. But then we have to have a cell to develop. And so one of the primary theories, or one scenario about a cell forming, is that a bubble formed around compounds. Here's this bubble formed around these compounds, and these compounds had some proteins and some genetic material in them. And this bubble then, lipids would form around this bubble, which would ultimately bond to form a membrane. And so that's a theory about the first membrane, sometimes called the protobiont is what that's sometimes referred to. Also referred to as cooperation. So cooperative processes producing this first membrane-bound molecule. All right, so that's kind of our theory about the early Earth atmosphere, and different possible scenarios about the first life forming. But then we have theories that move on past that, that look into how we got to where we are today. And so that's looking at kind of the historical ideas about biodiversity. And that's where um, macroevolution, theories of macroevolution come in. Now, macroevolution is sometimes explained in like family trees. This is called a phylogenetic tree. So phylogeny, or sometimes phylogenetics, is the study of organisms and their genetic to determine um, relationships with each other. All right, so talking about past relationships, how they're connected. So, like you would if you were going to do a family tree of you and your family. Those are connections. So, and we put trees together, and they're called phylogenetic trees. Now, generally, it's based off of physical appearances, things that look alike. Like right here, we have, in this case, we have a coyote and a domestic dog, and they look very similar. So we're going to say they look very similar, but we can also go into the genetic similarities. That's becoming more popular, comparing that, which is just the code for the physical traits, basically. So they should be very similar. But so we have this connection that we can make here. The skunk and the otter here have similar body plans, and so we'll put them here, and the cats over here. Cats are just unusual. They're over here by themselves. So we have these on the family tree. They're all carnivores, so we put those in the same order there, but different families. Now, these two would be in the same family, but a different genus, and these are going to be in the same genus but different species. So, that's a phylogenetic tree, putting things together like that and making inferences about relationships with each other. Now, sometimes we have good evidence to support that these things have an ancestor in the past, and sometimes we don't. Generally, we're talking about fossils if we're talking about in the far distant past. So, putting fossils and trying to make evidence from fossil record is very incomplete. We don't have all the fossils of everything that ever died, so we don't always have a good picture. And so a lot of inference has to go into some of these family trees. So that's phylogenetics. Something very similar is called cladistics. This is a field of still looking at the branching and common relationships in organisms, but it takes specific types of traits and puts those on the branches. For instance, these four animals here, we're going to put those on different branches of this tree because of these particular traits. Now, they all have vertebral column, they're all vertebrates, so they all make it past the first clade. That's called a clade. Now, they all have a vertebral column, but the turtle does not have hair and mammary glands like all of the rest, so it falls off of the tree right here at the first clade. The platypus has vertebral column, hair, mammary glands, but it does not go through gestation. It lays eggs. It's an egg-laying mammal. It branches off here, and then the kangaroo branches off before it gets to long gestation. So the beaver has all these traits. It makes it to the end of the cladogram. So it's the same type thing as a phylogenetic tree, but we have these very specific traits that show that divergence. So it's putting these animals really on here based on their physical characteristics, and that's what these are developed for. Okay, so we're going to pick up and continue with this lecture in the next podcast, talking about the historical look at geological past with evidence of fossils and things like that. So we'll pick that up in the next lecture.